Thank you so much to Darcy Failings for that incredibly kind introduction. Just making sure everyone can see my slides, which looks good. Okay. Well, I am honored to be giving this talk today on the pathophysiology of dystonia and cerebral palsy on this important day, National Day of Truth and Reconciliation. I have no relevant disclosures. I wanted to start out by talking about the ethos of our lab. We're a research lab focused on dystonia in cerebral palsy. And our general conceptualization of this is if we're going to study the pathophysiology of dystonia and cerebral palsy in animal models, we first have to optimize how we think about it in people. So to do this, we create standardized video registries of people with cerebral palsy. We ask experts in movement and cerebral palsy, and that's people directly affected by CP, caregivers, people with CP, and clinicians to describe the movements they see in these videos. We compare these features that these experts describe between people with and without dystonia and with different dystonia severities. And we use these features to quantify dystonia in those same videos using pose estimation techniques and machine learning techniques. This allows us to directly translate what we're calling clinically derived features that let us quantify dystonia to mice, to animal models. And we develop mouse models of neonatal brain injury that we think are translationally relevant and represent key features of clinical features of cerebral palsy. We modify the activity of different targeted neuronal populations in these mouse models using systems neuroscience techniques like chemogenetics and optogenetics. And then we use these techniques combined with these clinically derived dystonic features to identify treatment targets for dystonia in cerebral palsy. So the overall work that we do is translating community and clinician expertise to translationally relevant markers of dystonia that we can then use to study the pathophysiology of dystonia and cerebral palsy. So I will be splitting up this concept into three parts. First, what is cerebral palsy? What do I mean when I say that? Second, what do I mean when I talk about translationally relevant quantification of dystonia? And how does that help inform our study of the pathophysiology of dystonia and CP? So first, what is CP? This is the 2006 definition of cerebral palsy. It remains the most commonly cited definition in the modern literature. And in it, cerebral palsy is described as a group of permanent disorders of the development of movement and posture, causing activity limitation, attributed to non-progressive disturbances in the developing fetal or infant brain. Now, as medicine has evolved, what we found in a lot of our work talking to clinician experts in CP in the US and Canada is that the majority of these clinicians have uncertainties with how to practically apply this definition. And these uncertainties center in a few domains. One is how do you conceptualize fetal or infant in a practical way? What is the oldest age at which someone can sustain an injury and still meet criteria for a cerebral palsy diagnosis? How early can you diagnose CP? The second domain is how to practically apply non-progressive, particularly in the setting of genetic etiologies that have variable genotype-phenotype correlations, some of which may be progressive or neurodegenerative, some of which may not. And also given the fact that people with CP are at a higher probability of developing neurodegenerative conditions in adulthood. So how do you conceptualize CP as a non-progressive or non-degenerative condition given these clinical caveats? And lastly, a minority of clinicians were uncertain about whether hypotonia was included as a part of the cerebral palsy phenotype. What clinicians were certain about, though, was that cerebral palsy is not defined radiographically. And this is an important point because it ensures that cerebral palsy remains a globally inclusive diagnosis, given that radiographic criteria for CP can be precluded by access to imaging, lack of standardization across imaging, across centers, and lack of access to standardized neuroradiologic interpretation. So the clinicians that we talked to across the US and Canada were firm on this. Now, given the evolution of care for cerebral palsy, the evolution of the conceptualization of CP, There is a new proposed updated description put out just earlier this year by Bernard Dan and colleagues. I'm not going to read this one, but it's here for your reference. What I would say is that the authors are very clear to call this as a description and not as a definition. And that's incredibly important because this is a general overview of how cerebral palsy may present in many people that carry that label. But what the authors were explicit about is that this does not explicitly address how cerebral palsy can be measured or include the practical aspects of cerebral palsy diagnosis, so the procedural elements for diagnosis. And as such, as the authors acknowledge, many of the uncertainties from the 2006 definition remain, including age of diagnosis, conceptualization of CP as not degenerative or not progressive. That same sort of tension remains in the description. Whether hypotonia is included as a part of the phenotype, noting that hypotonia, as the authors acknowledge, is an early manifestation of CP, and several CP registers internationally still include individuals with hypotonia alone as part of the CP umbrella. And there's also a description of neuroimaging findings of CP, which I've already described. The vast majority of clinicians agree that this limits the inclusive nature of a CP diagnosis. So reliance on this as a defining feature remains difficult. So with this in mind, there are a group of people, including Darcy Failings, who so graciously introduced me, that have been working on an evidence-based, standardized, and most importantly, practical guide for how to diagnose cerebral palsy based on the vast body of evidence that we have available to us to do this in a standardized and reliable way. So what we've published as sort of a draft framework thus far is that cerebral palsy is a lifelong neurodevelopmental condition that affects functioning, where atypical brain development causes motor signs before age 2, and these are motor findings that you can see on a neurologic exam, without loss of motor skills by age 5. Now, this is a draft diagnostic framework still. We are looking forward to shoring it up with a systematic synthesis of the available evidence and really rigorous consensus building procedures. So watch this space. I'm really excited to be in this field now more than ever. I think that there is a lot of synergy that is going forward with both a practical, standardized, evidence-based diagnostic framework and the descriptions that have already been made available to us for decades at this point. So now that we've talked about cerebral palsy, I would like to talk about how we think about dystonia and CP and how we're doing that a little bit differently in the lab. So this is an aggregate of the consensus definitions for dystonia. Dystonia is voluntary movement triggered overflow muscle activation that's arousal dependent. So when I talk to families about dystonia, I say that it's every time you try to move or every time someone tries to move you or handles you, a part of your body that you did not intend to move moves out of your control. So inherently, dystonia can be associated with functional limitation, even just by that definition. What we do know is that dystonia is commonly associated with pain, commonly associated with gross motor functional limitation, that the data that we have for treatments are really limited, and that we have even less data on new treatment targets for dystonia specific to cerebral palsy. Yet dystonia is quite common in people with CP. Somewhere between 70 to 80% of people with CP have some degree of dystonia. Yet despite all this, we still struggle to identify and assess dystonia clinically. And in one of our studies, we saw that pediatric movement disorders physicians specializing in CP only diagnosed leg dystonia in 13% of their own patients, compared to their own later out-of-clinic assessments of their videos. So these are experts. These are pediatric movement disorders physicians who have dedicated their careers to caring for people with cerebral palsy. So I tell you this because dystonia, the difficulty in clinical dystonia assessment and underdiagnosis is not an expertise problem. It is very clearly, to me, a systems problem. And we've developed a variety of ways to assess this and to address this. I'm going to be talking about one of those ways today. The way that one of the ways we've thought about it is moving from this very general definition of dystonia across limbs, across etiologies, to thinking about dystonia specifically in CP and specifically in the legs, which is the most common site of dystonia in people with CP. And what our studies have shown is that in this context, dystonia can be quantified by looking at leg adduction variability and leg adduction amplitude, maybe more so variability than amplitude. So how we went about that was thinking about what is current gold standard dystonia diagnosis. And if you were to ask a pediatric movement disorders physician, which I am, it is expert consensus. So it's a group of dystonia experts sitting in a room, looking at a video or assessing a person together, talking with each other, talking about their feelings about movements, and coming out with a dystonia assessment. But as I've already mentioned to you, this is incredibly difficult to generalize to even those same experts when they're operating in isolation in a clinic, let alone someone without targeted dystonia expertise. So what we tried to do is quantify this expert consensus expertise using a couple of language analysis methodologies. So we asked experts to first justify the presence of dystonia. We looked at a ranking of the frequency at which they mentioned certain ideas in their consensus building discussion. And we saw that the presence of variability was a commonly cited feature that experts used to justify the presence of dystonia in the legs, and that the absence of dystonia was commonly used by experts to justify the absence of dystonia in the legs. And this was while reviewing videos of young people with cerebral palsy walking. We then asked experts to describe how they assess dystonia severity. One of the most common feature they used was this concept of adduction variability. So how variably are the legs going in and out during gait? And here, this is a breakdown of how often they described adduction variability with regards to their own qualitative assessments of dystonia severity. So the darker colors here are more severe dystonia by expert consensus assessment. And what you can see here is that they more commonly said that leg adduction was variable with more severe dystonia. And they more commonly said leg adduction was not variable with less severe dystonia. The second most common thing that experts cited was adduction amplitude. And similar to what I described with variability, again here, the darker colors represent more severe dystonia as assessed qualitatively by expert consensus. Experts were more likely to say that adduction amplitude was small with less severe dystonia, and more likely to say adduction amplitude was large with more severe dystonia. So we took these features that we'd identified from, in this case, clinician expert consensus, and we looked at the videos that they themselves are analyzing, and we used pose estimation techniques to develop gait variable measures of these expert cited features using nothing more sophisticated than a smartphone camera video that we took during our clinical assessments. And we looked at a few different tasks to try and quantify leg adduction variability and amplitude. One is gait, and the other is a seated task because not everybody with cerebral palsy gets around by walking. So we wanted to make sure that we looked at multiple different tasks when we assessed leg dystonia. What we saw during gait was here on the x-axis, increasing values indicate more severe dystonia based on expert qualitative rankings. On the y-axis is the degree of leg adduction. So smaller values on here means that the legs are closer together or more adducted. And there was no significant correlation between the amplitude of leg adduction during gait and expert-rated dystonia severity. But there was for adduction variability during gait. There was a positive correlation. More adduction variability, higher values on the y-axis was associated with greater dystonia severity on the x-axis. We saw similar things when we looked at the seated tasks. So during the seated tasks, the legs, the closer the legs got together during the seated task, the more severe the expert rated dystonia. Although the R-squared values were a little bit on the lower side. But when we looked at adduction variability in the legs, the more adduction variability, the more severe the expert rated dystonia with higher R-squared values. And what we were able to do is additionally show that this adduction variability and amplitude were specific to dystonia because, as many of you may know, people with cerebral palsy commonly have both dystonia and spasticity. In fact, that's the most common presentation that we see in people with cerebral palsy. So these features of adduction variability and dystonia, with variability shown here, were not associated with spasticity severity and were specific to dystonia severity. What we then did was use these features to train machine learning models to develop software called dystonia. This is work from my postdoc, Rishabh Bajpai. This paper is in revision, but the software is freely available to use. So please scan this QR code. It is available for use for any seated hand open close video. I know Rishabh is very eager to help folks optimize this. So please reach out to us if you have any questions about how to use or implement this at your centers. In developing dystonia, Rishabh also found that although adduction amplitude and variability metrics were in aggregate useful for discriminating during a seated hand open close task, when using something called feature selection methods, you can rank these features based on the importance or the degree to which they contribute to these machine learning models. And what he found was that the variance features had a significantly higher importance than the amplitude features, again suggesting that adduction variability, maybe more so than adduction amplitude, is contributing to how we can discriminate or assess for leg dystonia in people with cerebral palsy. So that was how we thought about quantifying dystonia in people. How do we apply this to animal models? The canonical way to assess dystonia in an animal model is with something called a tail suspension task, where you hang a mouse by its tail and look to see what it does with its hind limbs. And this model of DYT1, which is a rare monogenic dystonia, shows the classic phenotype that has been called dystonia in mice, which is clasping. Now, if you're thinking tail suspension isn't a very clinically relevant task, you're absolutely correct. But what tail suspension does highlight is a task where the mouse should not be bringing its legs together. The legs should not be involved in this task. And it's a task that involves handling the mouse. So if you think about when if you were to be a caregiver of someone with dystonia and you were transferring them, or you were being transferred, and your legs cross or scissor, that in effect is something very similar to what may be mimicked in this assessment. And what is clasping, if not the most severe form of leg adduction? And we can actually quantify this in mice. We can see that if we quantify the adduction, so how close the legs are together in these mice, that whether they're clasping or not, we can see that there is those legs are held closer together in a genetic mouse model of dystonia compared to a wild type mouse. So now that we've thought about translationally quantifying dystonia, both quantifying it very clearly in people and then translating that across species, how does that help inform our understanding of dystonia pathophysiology? So dystonia pathophysiology has most commonly focused on deep gray matter injury, particularly at the striatum. This is a meta-analysis we published years ago showing that in the published literature, imaging studies of children with dystonia and CP had largely demonstrated that the lentiform nuclei, namely the putamen, tended to have the most burden of injury. Later on, we published that if you look at babies that have been cooled after having hypoxic ischemic encephalopathy at term gestation, so these are children at high probability of developing CP, babies that had higher diffusion restriction or higher injury in the putamen were more likely to develop dystonia than babies that did not have that injury. And then in our mouse model of term HIE, we showed that there's actually an increase in the number of striatal cholinergic interneurons in the striatum, and that's selective to this interneuron population in the striatum. It's not true for other interneuron populations. And this is an important finding because we know in monogenic dystonia etiologies, the dystonia is associated with abnormal excitation of striatal cholinergic interneurons. So we know that this cholinergic interneuron excess activity and number is associated with dystonia, but is it actually causative? We also know that there's typically a delay to seeing dystonia after injury. So sometimes that can be months after neonatal brain injury. In people after a stroke, it can be years. Now, is this just a fluke, a neurodevelopmental phenomenon, or is delay actually necessary in order to generate dystonia after an injury? Does the underlying circuit pathophysiology have to persist for a long period of time before dystonia sets in? So we examined this in mice using chemogenetic techniques. We looked at that same tail suspension task, and we looked at measures of leg adduction amplitude and variability with chronic excitation of striatal cholinergic interneurons. Those are the interneurons that were increased in number after a term HIE in mice. Those are the neurons that are abnormally excited in genetic models of dystonia. What we saw was that after over 14 days of excitation, there was no difference in leg adduction amplitude in these mice, but there was a difference in leg adduction variability, although that difference only occurred at day 14, not acutely, not on day 1 of excitation, meaning that there did have to be a period of chronic excitation in order for that dystonic analog to set in these mice. We also saw that if we gave these mice artane, that variability, that adduction variability went away. We started giving these mice artane early, right as we started exciting these cholinergic interneurons. And this might suggest that if we can intervene early in people that are at high probability of developing dystonia, it's possible that we can thwart this delay-mediated phenomenon after injury of developing dystonia. So this supports a lot of clinical literature in deep brain stimulation that also suggests that early intervention may be more successful than delayed intervention for addressing dystonia in genetic dystonia etiologies and in cerebral palsy. But is the striatum the whole story? So we have all seen many people with cerebral palsy, genetic or otherwise, that do not have striatal injury that still develop dystonia. And we quantified this in one of our studies that of people with CP and isolated PVL, periventricular leukomalacia on MRI, so that's only cortical injury, that the majority still had some degree of dystonia. And when we roughly quantified region of interest size and injury burden using something called the Kitakoro criteria, which is a longstanding criteria for assessing neonatal brains, we didn't see that deep gray matter size or injury was associated with dystonia in babies born preterm. It was interhemispheric distance, so a marker that more represents cortical atrophy than it does deep gray matter injury. And there's already a plethora of evidence in adults with idiopathic dystonias that abnormal inhibition of the sensory motor cortex is associated with dystonia. So what we wanted to know is this actually a causative phenomenon, and can we identify this in mouse models of preterm birth? So we developed a mouse model of preterm birth where we inject the mice with mifepristone at a very strict time. These are time-bred mice. We film all of our deliveries, so we know exactly how long gestation is. We know exactly to within 15 minutes when the pups are born. We're able to induce birth and generate live pups up to a day early, which is quite a bit early when considering the human equivalent gestation. It's about 2 weeks early. Every day is about 2 weeks of human neurodevelopment. And then we assess the gait of these mice when they become adults. So what we find is that when we look at adduction amplitude on a treadmill, so this is a treadmill that we built to assess dystonic features that we think are clinically relevant based on our clinical work. As we increase the speed on the treadmill, which is what you see on the x-axis, the mice that are born preterm in teal are holding their legs closer together. So those feet are turned in. We also see that that turning in is more variable in these mice born preterm, again, mimicking what we might see clinically or what we've quantified clinically in young people with cerebral palsy. What we see in the sensory motor cortex in these mice is we specifically were looking at inhibitory interneurons, parvalbumin positive interneurons in the cortex. These are the interneurons that are primarily responsible for inhibitory drive in the sensory motor cortex. And we saw that in preterm mice, they lose these neurons, so reduced number of parvalbumin positive inhibitory interneurons in the sensory motor cortex, and reduced staining overall, which could either be due to reduced number or reduced arborization of these neurons. And we see that this is selective to the sensory motor cortex. So when we look in the striatum, for example, there's no significant difference in the number of parvalbumin interneurons. So what we see is that perhaps reduced number or activity of parvalbumin positive GABAergic interneurons, these inhibitory interneurons in the sensory motor cortex, are associated with dystonia, but could this be causative? So what we did was repeat some of our chemogenetic experiments, but we inhibited specifically this neuronal population, sensory motor parvalbumin positive interneurons. And what we saw during the tail suspension assay was that the mice held their legs closer together and that they did so more variably. So this, we really felt, replicated those clinical metrics that we'd identified. We also felt that we couldn't do these experiments if we'd relied on clasping alone. I will be honest and say that none of our mice, none of our preterm born mice, none of our mice that have neonatal HIE, none of the mice that have had the parvalbumin or cholinergic interneuron excitation, none of them clasp. But they do have this variable adduction amplitude that is representative of what we see clinically. So this, to me, really highlights the value of that translational work, going from clinical dystonia quantification to mice to hopefully develop the best translationally relevant research targets. What we're investigating now in these preterm mice is this concept of cortical dystonia of prematurity. There's been a lot of focus on treatment targets and targeting pathophysiology in the striatum and deep gray matter. We really focused on the cortex. And given that most people in the US have CP primarily with prematurity as the factor that's predisposing them to CP, with many having cortical injury, we really feel like this is a key feature to focus on. So I've talked to you today about cerebral palsy, how we can think about moving from a description to adding a practical diagnostic framework for use in clinics that's both evidence-based and standardized, how we quantify dystonia and cerebral palsy, and how that can be represented in the legs in young people with cerebral palsy by adduction amplitude, and perhaps more than that, adduction variability, and how that informs the pathophysiology of dystonia and cerebral palsy, not just focusing on the deep gray matter or the striatum, but also thinking about the cortex and cortical treatment targets. With that, I would like to thank my lovely and obviously very loving lab, and I am happy to take questions. Thank you so much.